Hello, this is the second lecture in the cardiovascular unit. In it we will talk about the action potentials in the heart, but in this first segment we will start with a review of microscopic anatomy. Please continue reading chapter 18. You may recall from the first term that muscle tissues in the human body consist of three different categories: cardiac muscle, skeletal muscle, and smooth muscle. So of course we will focus our attention at the cardiac muscle. The cells in this tissue are called cardiomyocytes. They're large cells that branch in multiple directions. That's kind of unique. They have striations, which means that contractile elements, actin and myosin, thick and thin filaments, are organized in the cell, and therefore it develops a large force in one direction, right? Cells are not as large as they are in skeletal muscles, and therefore they can be served by a single nucleus. Also, the cell predominantly operates on aerobic glycolysis. Therefore, the cell is very, very rich in mitochondria, myoglobin, which is a protein that stores oxygen, and that cell again predominantly produces ATP for muscular contraction using oxygen and using aerobic breakdown of glucose. Therefore, the myocardium has lots of vessels, heavy vascularized. Okay? And finally, cells are connected via the intercalated discs. On these two images, you can see a section through the cardiac muscle, and this is a relatively large magnification, so you can see individual cells. So cells are striated on both images, which means that thick filaments, represented by darker lines, and thin filaments, which are represented by lighter lines, are organized in a very specific manner. You can also see that cells have large nuclei. Here's another one, and here's a nucleus, and nucleus again. You see how those are large nuclei located in the center of the cell, one per cell. Cells are branching. Okay, here they are. And finally, the connection between the cells is pretty dark on both images. Those are called intercalated discs. I know we talked about intercalated discs last term, but perhaps it's a good idea to refresh. Intercalated discs are those invagination when one cell kind of protrudes into another. On the cross section, here's the end of one cell, and you can see the finger-like projections. So each of those are called intercalated disc that actually contains several types of intercellular junctions. The two types are desmosomes, which are very much like a rubber band that stretch from a membrane of one cell to another. It allows the two cells move past each other but not too far. Very much like if you were to tie a rubber band between your fingers, you can still move your fingers past each other, but eventually the rubber band will bring them back into their original position, right? So that allows the cells in a heart to hold on each other when they're contracting. Remember, in skeletal muscles, muscle is attached to bones, and so when the muscle contracts, the two bones are moved closer together. Well, heart's not attached to any bones, so when individual cells contract, for the whole chamber to decrease in size and produce this pumping effect, the cells need to be connected, and that's what desmosomes do for us. Gap junctions are basically pores that lead from one cell into another. And what that type of junction accomplishes is a flow of ions from one cell into another. And as you recall from an electrophysiology part from last term, ions carry charge and therefore change in voltage. So that basically means that if the cell on this side becomes excited, flowing charge or flowing ions through the gap junction will result in exciting the neighboring cell. That is very, very important feature in the heart, because cells then function together as what we call functional syncytium. It basically means that cells do not contract one at a time. They contract as one humongous cell, and therefore they can develop this huge force and pump the blood out. What we have learned so far is that in cardiac muscle, impulse spreads from cell to cell without any obstructions, whereas, as you may recall, in skeletal muscles, cells are electrically isolated from each other. So for each cell to get excited and then contract, each cell needs to be stimulated by a nerve ending independently. So in cardiac muscle, because ions can flow from cell to cell, this excitation spreads from cell to cell, and the whole heart works as a functional syncytium. A small animation that deals with microscopic anatomy of the heart is to follow.
The myocardium is comprised of cardiac myocyte and non-myocyte cells. Cardiac myocytes make up approximately 70% of the mass of the heart and are enmeshed in a collagen network that acts as a supporting framework. Blood is supplied to individual cardiac myocytes through capillaries that are in close proximity to them. Cardiac myocytes are comprised of actin and myosin myofilaments, whose organization gives rise to the characteristic striated appearance of cardiac muscle.